Hello, everybody. My name is now Tsuchiya. I'm going to be the chair of this session, 1S09M, Understanding the Relationship Between Structures of Conscious Experience, or Qualia, and the Structure of Information. So before starting this opening remark, I want to remind you that all contents will be recorded and talk parts will be uploaded to YouTube later. And discussions, Q&A will not be uploaded, so be assured. And please ask questions either using Zoom chat function or raising a hand or a Q&A. Or if you're at the conference room, then please come to the mic at the conference center. So this is the overview of our symposium. As an organizer, myself, now Tsuchiya, Monash University, and Marieke, Western University, Canada. And the three speakers, Makiko, Yamada, Masafumi, Oizumi, and Hayato, Saigo. So here's the preview of the program. Initially, we'll have two talks on neuroimaging by Makiko and also Marieke, followed by question and discussion. So we'll take all the questions at once in this 15 minutes time, not right after Makiko's talk. And then followed by that, there will be information theoretic or mathematical kind of talks by Oizumi and also Saigo. Again, and followed by another discussion. Finally, I'm going to give a talk on psychophysical approach to Qualia structure, followed by discussion and the closing remarks. So just to give you a kind of an overview of what this symposium is all about, and it is founded or supported by Transformative Research Areas B, Qualia structure. And if you are interested, please try to find search Qualia structure or Qualia kozo in Japanese. And me and Makiko and Masafumi and Hayato are part of these teams. And in addition to us, we have also members, I and Steve and Yusuke. And as a part of this Qualia structure project, we already had information structure of brain and Qualia symposium earlier this year, where Shinji Nishimoto and also Marieke gave a great talk. And if you're interested, you can see the symposium over there through the YouTube. And not only that, but also we had several other interesting symposium and talks on the neural base of consciousness channel. So if you're interested, please subscribe. And as another symposium, we are planning to have a development of consciousness towards the end of the year, mainly organized by Yusuke. So just to give you a summary of Qualia structure project and also what this today's talk is about. First thing that you will hear is that brain imaging kind of approach. And there, Makiko today is going to talk about measuring and manipulating the brain while we are asking the sort of Qualia experience consciousness in subjects while they're in the scanner. And then Masafumi and also Hayato will be talking about how to extract relevant information about our conscious experience through information theory or mathematical theory called category theory. And then in the end, I'll wrap up with sort of a new way of approaching this problem of how to characterize our conscious experience, Qualia structure, through a massive report paradigm. And that's the link between these three approaches, neuroimaging, mathematics, and psychophysics, new types of psychophysics, is going to be the one that we are going to pursue. And just a little bit of the advertisement. As a part of our project, we are trying to kind of push this idea or terminology of a Qualia through the general public. And if you're interested, or if you're children are seeking for some kind of summer project, then please consider this mascot character that you see in my background of this Zoom. And also we are also looking for sound that we can use for our project, and also to think about sound Qualia. If you're interested, please go to this Twitter website or homepage, please. All right, without further ado, I'm going to hand it to Makiko. Makiko, please share your slides.
Yes.
Okay, so the first talk of our symposium is by Makiko Yamada in quantum science technology. And the title of the talk is Brain Activity Measurements on Qualia Structure.
Okay, thank you for your introduction. I'm Makiko Yamada from QST Japan. I'm talking about brain activity measurements on qualia structure. This research is conducted under Transformative Research Area B, qualia structure. Here shows the conceptual framework of our group as now just introduced. Now it's going to extract the qualia structure from psychophysics experiments. I'm extracting qualia structure from brain activity using that same behavioral task under fMRI. And Dr. Oizumi is going to connect and find the mathematical relationship between the behavioral-based and brain-based qualia structures. My today's talk is mainly to introduce our plans of how we measure the brain activity on qualia structure. In order to extract brain activity corresponding to qualia structure, qualia similarity structure, we are using representational similarity analysis developed by Dr. Krieger-Skorte and Dr. Marr. Dr. Marr is the second speaker of this session, and she will present the method more deeply. So here I'm showing a very brief outline. Using fMRI, we measure brain activity patterns for each color and compute dissimilarity of every pairs by subtracting correlation across space from 1 here, and make the representational dissimilarity matrix. So the higher number indicates the brain activity patterns are different. And then we can see how they are arranged in 2D space using multidimensional scale construction method. And we call this spatial arrangement as qualia structure for now. Let me show you our task design specifically. These 9 color stimuli were obtained from HSV color space. Inside the MRI scanner, participants were presented 1 color disk and were asked to compare the similarity of presented color with the one in the previous trial and rate the similarity with 8 points. From 1 to 4 indicates the degree that they look similar, and from -1 to -4 indicates the degree that they look dissimilar. There are 82 trials in total. Here I am showing the pilot data obtained from 1 participant. By the way, the study was conducted by Takahiro in our group. Based on the similarity ratings, the dissimilarity matrix and the 2D arrangement were obtained as shown here. Black indicates that the pair was dissimilar, and white indicates that the pair was similar. As MDS indicates, the subjective similarity of colors are circularly arranged, which looks like HSV color space. In RSA analysis, representational similarity analysis of brain activity patterns, we analyzed the V4, which is known as color area. Here shows dissimilarity matrix and MDS. They look somewhat different from psychologically constructed space. So our next step is to find the mathematical law that transforms brain-based space into psychologically based space so that we can find the law of how the brain creates a subjective experience. This law is going to be the theory of consciousness, and this will be conducted by Dr. Oizumi, who is the third speaker of this symposium, and will explain the idea more deeply. In our brain activity measurement, we will also verify the causal relationship between qualia structure and brain activity by pharmacological manipulation. We are using mazindol. This is a catecholamine reuptake inhibitor, which binds to the dopamine transporter here and blocks dopamine reuptake. Therefore, it increases endogenous dopamine concentration in synapse. By measuring brain activity in placebo and mazindol conditions, we can test if the qualia structures are modified. But you may wonder if dopamine is related to visual experience, because most of the previous studies have shown that the dopamine plays pivotal roles in pleasure, reward, motivation, learning, and movement. But there are also evidence that visual experience is innervated by dopamine. I will show how our previous studies that indicate the relationship between dopamine and visual experience or consciousness. In order to measure dopamine in human brain, we are using positron emission tomography, PET. Using PET, we can quantify how much receptors are available in the specific areas in the brain. Here, I will introduce our studies that used a radioligand called raclopride, which is an antagonist on dopamine D2 receptors. Raclopride binds to D2 receptors in striatum here. We can quantify the amount of ligands that binds to receptors for each subject. Red area indicates high binding potential, which means high dopamine receptor availability. Using PET and fMRI, we have revealed that the dopamine D2 receptor availability in striatum is related to the brain activity of subjective experience of colorfulness. Inside MRI scanner, participants were presented 4 pictures sequentially. And after high saturation, middle saturation was presented. And after low saturation, middle saturation was again presented. The level of saturation of these 2 pictures are physically same, but subjective experience is different. The one after high saturation looks faded compared to the one presented after low saturation. This subjective difference was reflected in the frontal and parietal activities, which were related to D2 receptor availability in striatum. Thus, we speculate that manipulation of dopamine level in the brain will affect on the brain activity patterns of qualia structures. Next, I will give another example of a study that showed the relationship between the amount of dopamine released in the brain and the state of consciousness. Here, as mentioned previously, we used mazindol, which blocks dopamine reuptake and increases endogenous dopamine concentration in synapse. In this condition, we use raclopride PET so that we can measure how much raclopride binds to D2 receptors. Blocking of dopamine reuptake induces competitive inhibition of endogenous dopamine. Therefore, raclopride, shown in cyan color here, does not bind as much as in placebo conditions. Thus, by calculating the change of raclopride binding potential, we can quantify how much dopamine increased in striatum. In the left plot, the horizontal axis shows the change in raclopride binding potential, and the higher number indicates the larger increase of dopamine in striatum. The vertical axis shows the change in state of consciousness measured by the altered state of consciousness rating scale. This result indicates that the increase of dopamine in striatum causes the change in the state of consciousness. And this is going to be the last example of dopamine. In resting state fMRI, we can extract the dynamic changes of different network states. Please take a look at the top figure here. The horizontal axis shows the time points. And for instance, this phase was manifested by default mode network state, and this phase was language network state, and so on. Here, under Mazindo and placebo conditions, we have investigated the dynamic change of these 12 networks. In upper figure here, vertical axis indicates the proportion of each network in total time. Blue indicates Mazindo and red indicates placebo condition. And using hidden Markov model, we have identified two meta states. Meta 1 is composed by state 1 to 7, and meta 2 is composed with state 9 to 12. And here, this left matrix indicates that under Mazindo condition, the intercorrelation in meta state 1 is reduced, but the intercorrelation in meta state 2 is increased compared to placebo condition. And we examined which state among here is modulated by the amount of dopamine release in striatum using raclopride PET. And we found that the increase of dopamine release in striatum caused the increased occupancy of high-level vision network state here, this one. Therefore, after dopamine increases, a high-level vision network is more occupied during resting state. This network is possibly associated with visual thought because in the previous study, it was revealed that this network ICA component was related to visual thought reported after scanning. Thus, the dopamine influences on the occurrence of high-level visual network, which is associated with subjective visual experience. In summary, I have introduced how we measure the brain activity of qualia structure. First, I have shown that the brain activity corresponding to the qualia structure can be extracted using representational similarity analysis, but we are further interested in finding the mathematical law that transforms brain activity into subjective experience. Second, I have shown that dopamine plays a pivotal role in visual experience, and therefore we are interested in how the manipulation of dopamine release in the brain causes the alteration in qualia structure. This is acknowledgement, and the last experiment was conducted by Daisuke, and the first one was conducted by Takahiro, and these are group members, and these are members of Transformative Research Area B qualia structure. Thank you for your attention.
Thank you very much, Makiko. We have roughly like 1 minute for questions if you have. Is there any pressing question from the audience or panelist? Otherwise, maybe we can take all these questions at once at the end of the Marieke's talk. Sounds fine? Okay, let's move on then. Marieke, please share your slide. And also unmute yourself.
Yeah.
Yep.
All right. Yes.
Okay.
Can you see my screen?
Yes. Great. And now I want to introduce. So this is again the Symposium 1S09M, and this is a second talk. The speaker is Marieke. She's from Western University in Canada. And her talk title is Predicting Perceptual Representations from Brain Activity. Please.
All right, thank you. And thank you for your introduction and for inviting me to the symposium to give a talk and organize it together. So let's go into the content. So here, when we see a stimulus, then there's brain responses associated with that. And then also there is a perceptual experience associated with seeing, for example, this apple. And as also was explored in the previous talk, now what we are interested in is how these brain responses relate to the perceptual experience that we're having when we're seeing object images in my scenario. So the question here that I'm going to focus on today is whether we can predict perceptual representations from the brain activity. And for that, indeed, what we first need to do is measure the brain responses and characterize those. And then we can ask whether we can predict perceptual experience, which we also need to measure, of course. So let's start with the brain responses. So what we did is we presented this set of objects to human participants while they were in the fMRI scanner. And as you can see, the images consist of a wide range of categories of objects. So they include humans, both faces and bodies, also animals, faces and bodies, and then natural objects. So that includes fruits and also natural scenes, and man-made objects, including tools and houses. So we presented this to participants while they were in the scanner. They were viewing the object images, doing a task unrelated to the images. And we measured their brain activity, and specifically, we were interested in looking at high-level visual cortex, because that's a region that we know is involved in object perception and categorization. So in order to then characterize these object representations in high-level visual cortex, we used representational similarity analysis. So I'll explain indeed in brief how we exactly do this, but of course, I think the previous talk already helped for this understanding it. So what we do here is for each pair of images, we measure brain responses. So that's the response patterns here for fMRI. This is across voxels in a region of interest, for us, high-level visual cortex. And then we can compute the dissimilarity between these response patterns, for example, using 1 minus the correlation across space. And that indicates how different the brain reacts to these images. And we can store this measure now in a representational dissimilarity matrix, or RDM. And we do that specifically now for IT, which is inferior temporal cortex. So that's high-level visual cortex, as I mentioned before. And it's shown here in blue in a schematic of the brain. And that's a region that we know is involved in object perception. So what we can see here is that the stimuli are on the axes of this matrix. So let's put the stimuli now there. And you've already seen them, right? Here they're now smaller, but they're just arranged along the axes, organized according to category. And now I'm just going to show schematically how we build up this matrix. So what we do is we know that, well, there's lots of dissimilarities in the matrix, one for each pair of object images. So the diagonal, by definition, is 0 in this scenario, because that's the dissimilarity between a response pattern and itself. And then the matrix is symmetric about that diagonal of zeros. So here for these two faces, this would, for example, this is schematically showing where that dissimilarity would be. And as you can see, it's blue because the two faces in this example elicit similar response patterns, right, as you can see here. So now we can also do this for another object pair, for example, a face and a banana, and we can see that those elicit more different response patterns. That's the warmer color indicating that as well. And now what I'm going to do is I'm going to show actual data from the fMRI experiment that we did, and this is averaged across 4 human subjects. So what we can see now is that there's clearly some structure in this data, in the representation. And one thing that is immediately obvious is that there is this division between animates and inanimates. So whenever you have a pair of objects that straddles this category boundary, you get warmer colors, so more different response patterns. We also see a tight cluster of human faces, so they elicit quite similar response patterns in high-level visual cortex. Now we can also show the same representation in a lower dimensional space with multidimensional scaling. That was already explained, right? But here what we now see is the images and the distances between these images reflect their response pattern dissimilarities. So whenever two images are placed further apart, they elicited different response patterns. When they're placed closer together, they elicited more similar response patterns. If we color code the images, we can also see, again, the division between animate and inanimate objects and the cluster of human faces. Now, this is the brain activity and the representation of these images in high-level visual cortex. And the very helpful thing about RSA is that now we can also measure perception of the same object images, and then again create a representational dissimilarity matrix. And at this level, we can ask whether the brain representation actually predicts our perceived similarity of the same objects. So now the question is, how do we acquire those data for perception? So here it's shown as also patterns for the different images. That could be a pattern across perceptual features. But what we decided to do is to acquire similarity judgments, because we wanted to be agnostic as to which types of features people are using. And if we directly ask for the dissimilarity between objects, then we're capturing everything that people use in terms of their mental representation. So the way we did that was by asking people to arrange these objects in a circular arena using multi-object arrangement method that we developed. And it's quite efficient to acquire a lot of similarity judgments with that method. So people were simply seeing these object images, and we asked them to arrange the objects according to their similarity into the white arena, where again, distances now reflect how different people perceive the images to be. And as you can see, whenever you move an object into the arena, you have to think about its relationship to the objects that are already there. And so it's quite efficient because you're judging multiple similarities at once. And then we also have multiple arrangements. So they would get a next trial to get access to a higher dimensionality structure than just two, right? Because the screen is of course just 2 dimensions. But with multiple trials, we can assess a higher dimensional structure. This method is quite useful, and participants only needed about an hour to provide their similarity judgments for the entire set of objects. So now we're back here, and now we can ask the similarity judgments, what kind of structure do we see there in the perceived similarity of the objects? So we can fill that in here. This is an average of 16 human subjects. And here we can now see that we do see some similarities in structure. Clearly, there is also this cluster of human faces. And we see also that between animates and inanimates, they're also perceived as being more different, as we saw in the brain representation as well. We also see some differences. So the similarity judgments seem more categorical. They're more blocked, clustered. And we see some different clusters there. For example, you can see humans and animals, each of them are a cluster, and we didn't see that so clearly in the IT representation. So now, just to visualize this also with MDS, to make it somewhat more intuitive maybe, we can do that as well. And then we saw the IT representation before. Here we see now for the judgment. So you can indeed see that there's more stronger clustering. We also tested this, whether that was indeed significantly different between the two, and that is the case. And we see here in the middle some of the images that people actually found hard to interpret. This, for example, is the back of a human head, but it also looks like a coconut, right? So there were some, you can immediately see this. We asked them also to give us some descriptions of how they did this. What we see if we color code these images, we do see again that there is also similarities. So for example, the division between animates and inanimates is quite clearly present in both. And that's the warm versus the cool colors. So now we want to quantify this, and we then correlated in the matrix the dissimilarities from the brain representation and those from the perceptual representation based on the judgments. And what we can do now is that we can show basically model performance where the brain representation is a model for the judgments. And we can ask what's the correlation between the brain and the judgment RDM. And here what I'm drawing is the noise ceiling. So that indicates performance of the unknown true model given the noise in the judgment data. So that's the model that would really fully explain the judgments. So ideally we want to end up in the noise ceiling. Now IT is here. So what we see is that it is significantly correlated with the judgments, which means it does explain a significant amount of variance in the judgments. And that's the case not only for all of the objects together, but also for these categories, when we split it into different categories, it holds as well. So that's the list here. And this says that the IT object representation does predict perceived similarities. So objects that elicit similar response patterns in your brain are also, they tend to be perceived as more similar. And we did also show that the object representation in early visual cortex does not predict the perceived similarity, consistent with this idea that high-level visual cortex is more important for explaining our perceptual experience. So now what we can ask is, can we do better? Because we also see that we're not in the noise ceiling. So in order to address that question, we looked at a few other models. So one is semantic models. We expect that people are using some semantic information in their similarity judgments. So what we did is, for example, for this apple, we asked people to generate, this is a separate set of participants, labels that describe the object features, for example, these ones, and the categories that this object belongs to, for example, these ones. And then what we did is we asked whether we can predict the judgments using those labels. And that will help us understand whether it's features or categories that contribute more strongly, and also whether that works better than the brain representation. And computational models, we also looked at a deep convolutional neural network. So this is a model of the visual system, which is quite attractive because it's image computable. You can take an image and provide a category label as output. And it also achieves nowadays human level performance on object categorization. So it may be a good model of how the judgments are generated, how we perceive the objects. So this is just an example. This is AlexNet. It's a deep neural network and that we use. So it takes the image and then it provides an output that's a category label. And we can ask whether the internal representations of AlexNet can predict our perceived similarity of the objects. So now to answer, can we do better? We actually can. So these two models, so of course, interesting to know which they are. They're actually better at explaining the similarity judgments than the brain representation. And those were the categories and a combined model of both features and categories. So that's driven by the categories, this performance. And we'll get back to that in a moment. So then we also have these two. So AlexNet is the computational model and the features. And they do not as well as the categories, but they are all significantly correlated with the judgments. And then we can see if we compare these different models that indeed the categories do better than any other model. And then AlexNet, so that's the computational model, and the brain representation, they're both doing quite well and better than the features. So this is interesting because it suggests that the perceived similarity of objects is best predicted by category labels. However, the IT representation also does quite well. So just as a last point to get a better understanding of why the category labels may be performing better, we need to get back to the differences we noticed between the brain representation and the judgments. So one of them was that there seemed to be different category clusters in the judgments, or at least a few additional ones. And in order to investigate that further and quantify that, we looked at the proportion of total category-related variance explained by different category divisions. And I'm showing the results already here for IT and the judgments. And this red color indicates the animate-inanimate division, very strong in the brain and also quite present in the judgments. Face versus body, stronger in the brain. And then these additional divisions. So that's artificial versus natural, and also human versus non-human. And these clearly come in the judgments and are not very strongly present in the brain representation. We also performed some inference to test that. So here we're just seeing the same proportions, but now in a bar chart. So these were indeed significantly different. So what this seems to suggest is that the similarity judgments reflect IT, but they also transcend it because they're introducing these additional category divisions, which are human related. So that's the artificial natural and human non-human distinctions. So what this suggests, so we're now at the last slide, is that first of all, optic representations in inferior temporal cortex may give rise to high-level conscious judgments of optic similarity. So they may be, they're a potential neural substrate for our perceived, our experience when we're perceiving objects, visual objects. Also, the similarity judgments, they don't only reflect, but also transcend IT, as we just saw. So there's these additional category divisions. And that's quite interesting because it seems to suggest that in our brain, or at least the visual regions, we have maybe a representation of optic categories that have a long-standing ecological relevance, like faces versus bodies, animates versus inanimates. But these, let's say, somewhat newer divisions, if you think about the evolutionary progression, right, that are related to just being human or non-human, they come in the judgments and they're not so visible in the brain representation. So maybe other brain systems may adaptively read out or introduce category divisions that serve current behavioral goals. And here you can think more about prefrontal or parietal cortices. And that's something we've been looking into, but it's a bit too much to talk about today. But with that, I would like to end here. And if there's any questions, I'm happy to answer them.
Thank you very much. Okay, so now we are in the discussion Q&A time up until like another 13 minutes or so. So if you have any question, you can use Q&A box in the Zoom, or type in question in the chat box, or raise the hand, please, audience. And if I don't see it, then maybe I'll start, or maybe the panelists can also ask question first. Is there any question, Masafumi or Hayato? Otherwise, I have a lot of questions actually. Or maybe then I'll start. So Makiko showed pilot results of N=1 subject, and then we saw some kind of discordance between the MDS representation and B4 based color structure. Marieke, did you try to do something like that with your also IT based object,
color information?
No, in terms of the N=1, individual based sort of the MDS correspondence.
Yeah, so I did look a little bit. Maybe I should try to quickly pull up a figure from a paper. So I looked to some extent just visualizing the individual variability between subjects. And there was certainly some variability. And in terms of the judgments, what was interesting is that we, of course, didn't really know how participants would arrange these images and whether they would choose to use category information, which they ended up doing. But not everyone did. And there was especially one subject who also reported, because we did ask, how did you do it? Who reported that they used shape and color information. And they certainly got a different arrangement. So maybe I can quickly share my screen again. So this is a figure from a paper where we look at this. So these different blue dots are the subjects. And then the big one, let's say, is the similarity judgments that are averaged across subjects. But here you see the individual participants. And then you see this one over here. This subject was the one who arranged more based on low-level visual properties. And you can see we also had some models of the images based either on, for example, luminance or silhouette, so pretty low-level visual features. That subject is closer because this is an MDS arrangement of the representations. So that subject is closer to these low-level models. And then you can see that the other subjects they're closer to these brain regions here in red, specifically indeed IT, right, and also FFA. And as you can see, there's some variability between the subjects. This subject is somewhere a little also closer to model of V1, but overall, you do see some clustering based on whether it's the judgments or the brain region, so that's blue versus red, or some other models that are low-level models of the images, except for, yeah, this subject over here, who also, again, in the report did mention, I'm not using category, I'm using shape and color. Yeah. So I don't know if that helps, but this is what we did look at for the variability.
Yeah, I think this is great. Yeah, go ahead.
In your case, you ask subject to make a similarity judgment outside the scanner, and then inside the scanner, what do they do the same thing, or like?
That's a very good question. So they're doing a different thing. So outside the scanner, when they're making the judgments, they're explicitly judging the similarities between the objects. But inside the scanner, they were doing a task that was unrelated to the object. So they had to just detect when there was a change in the fixation cross had a color that changed when an image came up, either blue or green, and they had to just report that. So it is a different task, and there is some studies, I think, by Hans Optebeek's lab from Leuven, where they did ask also participants to make a judgment, a similarity judgment, more in terms of when they had to judge the similarity to the previous image when they were watching or seeing images in the fMRI scanner. And I think they did find something relatively similar to what we find, which seems to indicate, together with some other evidence, which is when you change the task in the scanner to, for example, make category judgments. We also tried this in a few pilot subjects. It doesn't change the representation much. So it seems to be a relatively default representation. And in the scanner, it's leading, whether you do a different categorization or you do a task that's unrelated to the images, or one that is related and judging similarity, it's relatively similar for these types of objects. Because we also thought maybe it changes quite a bit if we change the task, but I think this happens potentially somewhere else and more in the readout of information, and the visual representation seems to be relatively stable. We also found this similarity, right, between the monkey representation. If we look in monkeys with electrophysiology recordings for the same images, also doing a task unrelated to the images, and humans, we also find that is quite similar, those representations. And then the one in the humans doesn't seem to be affected too much by the task you perform on an individual image. So it seems quite stable. And the larger influences of task only start to happen when you have competing stimuli at the same time. And then you start to see effects, but not so much if there's an individual image. Yeah.
I see.
Thank you.
Okay. Is there any question from you, Marieke, to Makiko or Masafumi or Hayato? Unless otherwise, I might just ask another question.
Yeah, maybe that's good. I mean, what I found very interesting was these different colors, so colored spaces, right? In the beginning, you showed some of those. I thought that was quite interesting, and I was wondering maybe if you know, in terms of subjects' experience of those stimuli, was that something that varied also if you changed? So did you find effects of task? I guess that's my question. Whether you played around with that, that people would do different, perform different tasks on these stimuli, would you find effects there in terms of their perceived similarity or brain-based similarity?
Actually, we haven't done that yet. We just did this experiment for one time. So, yeah, we have to take a look after that.
So that's kind of our experiment related to that. And we haven't done that by ourselves, but in the past, it seems like when you ask participants to pay attention to the color structure, like, you know, red and green supposed to be opposite, and blue and yellow are supposed to be opposite. If you tell them, then they start to change the judgment, it seems.
Yeah, exactly. Yeah, yeah.
But
we saw that too.
We haven't done that.
With the judgment, they're quite flexible because that they change, but then the brain representation in the visual region doesn't change so much. Yeah. Yeah.
So what do you think will be changing is probably like higher level areas like parietal or
exactly.
prefrontal.
Yeah, that's what I think. And I think what you would find there, which is something I've looked at in a separate experiment, is that you would find a representation in those areas of the rule. So are you categorizing based on color versus shape, for example, or category? That you can then find sort of what rule are they using. But you can't really see so much that the representation of the objects changes. And that may be because of multiple reasons. It may just be that fMRI may not be the best method for measuring these dynamic representations in frontal and parietal regions. Yeah, I do think that's where it's happening. And it is more of a readout. So of course, all the visual information is there. It's just how do you read it out and combine it with what you're supposed to be doing right now for the task. And I do think, yeah, indeed, that's much more, that goes beyond visual cortex.
I see. Yeah. And then that kind of leads to another sort of my question, if there is no other pressing question, if it's okay with you guys. So it's also related to probably the talk, next talk, Masafumi's or Hayato's, although I don't know what they're going to talk exactly. And also Makiko's another interest of the quantum cognition. So so far, both of you, Makiko and also Marieke, you are assuming that if you show A and then B, you say similarity of 1. And then if you show B first and then show A, then the similarity will be also 1. Right? But in reality, in our experiments, especially when things are kind of close together, similarity judgment can also change. And also, like, the case of is China similar to Korea, or is Korea similar to China? It's known to flip a bit. So based on your kind of, conceptual kind of, mapping of the object similarity, or maybe it's actually reflecting the perceptual similarity, if this kind of asymmetry happens in terms of similarity, would you predict that that should be happening in the level of the visual cortex or at the level of the conceptual readout or rule-based kind of thing in the prefrontal, or is it kind of the emerging feature? What do you think?
Yeah, that's a good question. I think my guess would be that it may depend on the type of feature that may be dominating the judgments. So if it is a lower level visual feature, I could imagine this to be visible in the visual cortex, thinking about neuronal adaptation, right? fMRI repetition effects. You may be able to see something there. And I could imagine that that would be the case. If it's a bit higher level, if it's more about a particular category membership, or you're judging some more abstract properties, then I'm not so sure if it would be in visual cortex visible. And it seems like then it may be something, that is a little, it's more processed in higher level visual regions. Higher level brain regions, right? Like frontal parietal cortex. But that is just a little harder to capture because they don't have a very strong visual representation. So you can't easily see how the representation then changes, right? But I do think, it depends on the property that people are judging. Yeah.
Okay. Does anybody want to say anything on this issue of the asymmetry of the similarity, or like the noise of the similarity judgment? Also, Marieke's analysis kind of posited that there is a ceiling, noise ceiling, in terms of performance. That's probably coming from the assumption that is it within subject noise or across subject noise?
Across subject noise. Across subject.
Okay.
Yeah.
So,
and the judgments do have lower noise in general. So if you just look at replicability within an individual, then that's more replicable than the brain data. And that's just the measurement method. And then we also had to indeed, when we compared, for example, the judgments versus the brain representation in terms of those proportions of variance explained by the different category divisions, etc., we had to sort of equate the noise levels to make sure we had a fair comparison, because they are different. Yeah.
I see. Okay, so now, unfortunately, it's time to move on, and I'll hand the chair to Marieke from here on. And Masafumi, can you start sharing?
Yes.
All right, that's great.
Yeah. Am I sharing? That's right.
Yes.
Yep.
Yeah, perfect. So we have our next talk, and this is indeed by Masafumi Oizumi, and it's the title is Towards Elucidating the Mathematical Relationship Between Qualia and Brain Activity. And this is just to mention the talk number, right? So it's 1S09M and then talk number 3.
Yep. Hello, so my name is Masafumi Oizumi. I'm from the University of Tokyo. So today I'm going to talk about theories of consciousness. So currently several theories of consciousness have been proposed, but not much experimental validation has been done yet. So I'll discuss how we can empirically test the theories of consciousness and utilize the theory for consciousness researches. So first, let me explain the general goal for consciousness researches. So in conventional neuroscience, we try to find the relationship between stimuli S and neural activity R. That is the function F that links stimuli S and neural activity R. So in consciousness researches, we do not care about this, relationship. Rather, we try to review the relationship between the consciousness, subjective experience, and neural activity. So so this part. So the ultimate goal for consciousness researches is to find a mathematical relationship between qualia and neural activity. So here G is some mathematical transformation. And the purpose of a theory of consciousness is to provide a plausible candidate for G. But a serious problem here is if we consider this is some mathematical equation, quail has to be some quantity. So unfortunately, there has been yet no established methods for quantifying quail. To resolve this problem in our Qualia structure project, we propose a novel framework for quantifying quail. So let me briefly explain our framework. So if you are asked to explain your subjective feeling quail of redness of red, you will be in trouble and feel like you cannot explain it with any method. So people believe there seems no way to quantify a quail, and consciousness cannot be science. But by changing the way of thinking, we can notice there is some way. Actually, there is some way to quantify a quail. So we can actually quantify the relationship between a quail and other qualia. For example, that we can quantify the relationship between the redness, the quail of red and that of orange. So in this case, the relationship can be a similarity between the qualia. So in this case, red and orange are similar, but red and green are dissimilar, and also red and blue are dissimilar. And in this way, we can correct the similarity relationship between quail of red and other qualia. So by accumulating such many relationships, we can actually, it is actually practically same as quantifying let itself. So this idea is theoretically supported by Yoneda's lemma in category theory, which Dr. Saigo, the next speaker, will explain more. So we call these relationship between Qualia structure. So by using the idea of Qualia structure, we can, in principle, test theories of consciousness. So to test our theory, first we need to quantify Qualia structure, so similarity relationship between Qualia by psychophysical experiments. Then we need to also record neural activities corresponding to each quell. And from the neural activity data, we compute the structure of brain activity. And then finally, what we need to do is to check whether the Qualia structure and the structure of brain activity are isomorphic. That's our basic strategy. And the simplest idea is that activity pattern R itself can be isomorphic to Qualia structure. So this is the kind of simplest idea, but we may find that this simple idea doesn't work, doesn't fully explain the Qualia structure. In that case, we need some theory which transform this neural activity into some other space. And in this case, this transform neural activity is isomorphic to Qualia structure. So among the many possible candidates of transformation G, we can empirically find the mathematical relation G that is most similar to Qualia structure. That's the first objective we can think of from this new framework of Qualia structure. And then another important problem is about the place of consciousness. So we know that the brain activity generates consciousness, but we also know that not all brain activity is directly related to consciousness. So we need to identify the subregion in the brain that is directly related to Qualia. So the problem here is to find the subset in the whole brain activity that is the most similar to Qualia structure. So in this case, we use the A for subregion of the whole brain activity. And then what we need to test is which activity in subregion X or Y is more similar to Qualia structure. So we need to check many possible subregions in the whole brain. So in this case, X and Y, and then evaluate similarity between Qualia structure and the structure of brain activity. Then test which one is better. In this case, Y is more similar to Qualia structure. So to summarize so far, what we can possibly do with the idea of Qualia structure, first objective is to find the transformation, mathematical transformation G that most explains the Qualia structure among plausible candidates. And the second objective is to find the subregion A in the brain that most explains the Qualia structure. So the different theories predict different mathematical transformation or subregion A. So I'll particularly explain the integrated information theories prediction about G and A in this talk. So the hypothesis, the II integrated information is called IIT. So IIT's hypothesis is that this mathematical transformation G is information structure of brain activity. So information structure is some structure of causal relationship between neurons. And about the place of consciousness, IIT's hypothesis is that subregion A that corresponds to consciousness is complex, which is locally most integrated subsystem in the brain. I don't have time to explain the details about information structure and the complex, but I'll just briefly explain these concepts. So first, the information structure is some causal relationship between the neurons in the brain. So in this case, we quantify the cause and effect for this very simple system, 3-unit system, and we can quantify cause and effect relationship for all possible subsystems of this ABC system, and we can sort of visualize information structures in this way. So please refer to the paper if you are interested in the details. And we can quantify this information structure in real neural data. In this paper, we quantify the information structure in the human ECG data. So we have some way to quantify information structure. So we can test whether this information structure matches with Qualia structure. That can be a test of the IIT's prediction. And also about the place of consciousness, the IIT's hypothesis is that the complex locally most integrated subsystem corresponds to the place of consciousness. So we can also find this locally most integrated subsystem in the actual neural data. This has been done recently by Jun Kitazono, my postdoc, and this is the result for monkey cog data, and this is this is the result for mouse connectome data. So the main message here is that we have some way to extract the complexes, most integrated subsystems, and then we can potentially test the theory's prediction, by comparing the Qualia structure and also the structure of brain activity computed in the complex. That can be a test for the theory. So as I mentioned, there are many other theories of consciousness: global workspace theory, or integration inference theory, higher order thought theory, or predictive coding theory, and possibly many others. So, but since there has been no established methods for testing these theories in experiments, we have no idea which theory is the best. But with the idea of Qualia structure, we can, in principle, empirical test which theory, most explains the relationship between Qualia and brain activity. Unfortunately, not all theories of consciousness is written in mathematics. But if these things are, written in mathematics, we can, fairly compare the theory's prediction with the, by combining the neural recording and the, psychophysics experiments. So to summarize, so first, I want to say that the quantifying Qualia via the relationship between, Qualia would significantly advance consciousness researches. And with this novel method for quantifying qualia, we can test and compare various theories of consciousness. And this will provide a new research paradigm that actually connects theory and experiments. Yeah, that's it. Thank you.
Great, thank you. So actually, we're sort of out of time now. So I think what we should do is go to the next speaker, and then after that talk, we can have a discussion and have questions about both talks. The next speaker is Hayato Saigo.
Yes.
And hello.
Hello.
And the title of the talk is Categories for the Working Neuroscientist. So we can already see the slides. Yeah, that's perfect. So
Okay.
Just to quickly mention again the talk number, that is same symposium is 1S09M, and this is the 4th talk. So without further ado,
Okay.
I think we can start.
Okay. Hi, my name is Hayato Saigo, and this title is Category for the Working Neuroscientist. There is a very famous mathematical book on category theory. It's named Categories for the Working Mathematician. So here, there is no technical subtleties, only the conceptual. We only focus on the conceptual side of category theory. So, this is my self-introduction. My original research field is quantum field, mathematical structure of quantum fields. Quantum field theory is unification of relativity theory and quantum theory. And but it is very difficult. It is well known that this is very difficult to unite the relativity theory and the quantum theory. So we go to the generalization of relativity theory, quantum theory to solve the very special problem. And the category theory, which I introduce here, is in some sense generalized relativity theory. That is, the relativity theory concerns that some kind of representations and the relationship between representations. And the quantum theory can be generalized to the non-commutative probability theory. Maybe I talk a little about that, but we focus on the category theory. And by unification, I believe that it contributes to consciousness phenomenon qualia studies. Okay, so let's go to the introduction to the category theory. What is category? The category is a mathematical system consisting of something called object and something called arrows. And the arrows can be composed. So this kind of structure is, how to say, very universal. For example, if we consider the physical system and considering the all possible state as objects and the all possible state transition as arrows, then it makes some kind of category. So here we call this category of mobility. The point is that the state transition can be composed because from the A to B, we translate something, and B to C, the state transition, then this whole transition has some structure of state transitions. So in words, the category of mobility here, the very first example of category is objects as states, the arrows and state transitions. I believe this category is very important to this not only the physics, but also the consciousness studies. A very subtle point is that what is state? This is a very deep question, not only the philosophical, but also the scientific question, because state is, from mathematical side, state can be represented as a statistical law or the expectational function in mathematical sense, determined by interrelationship between a system focused on and the environment. Environment means some system significantly interacting with the system focused on. So the important thing is interrelationship is very important. And of course, the environment itself is a system, so we can consider the meta environment or something. But we go on the next slide. For the application to science, especially to the realm of neuroscience, it will be important to understand that interesting categories can be considered as a functor categories. I will introduce two important notions, that functors and natural transformations. As you know, as you understand later, of course the category itself is a very abstract thing, and objects, anything can be considered as object, and anything can be considered as arrows when it satisfies the axiom of the categories. But very interesting and concrete kind of categories is functor categories, whose objects are functors, and the arrows are natural transformation, which we introduced. And Professor Oizumi mentioned about the Yoneda lemma, but Yoneda lemma is all about this relationship between the abstract category and the functor category. Yoneda embedding is essentially say that every category can be seen as a subcategory. This is a subsystem of a category over a functor category. Actually, it has it is called a perceived category, and it has a structure of topos. But the Yoneda lemma topos, today we do not go on the topos, but the Yoneda lemma, the essential contents of Yoneda lemma is objects are isomorphic if and only if the corresponding functors are naturally equivalent. So we introduce the notion of the functors and the natural transformation and natural equivalence and so on. What is functor? The functor is, shortly speaking, the morphism mapping from the category to the another category, preserving structure of category. So let's say the C is some category, has object and arrows and compositions, and D another category. The functor is a mapping from C to D, preserving the composition structure, that is this equation, this FG composition, F equal FG composition FF. And this functor is a very generalization of the representation, very general notion of representation. Representation of something is the mapping from some region to the another region, preserving the structure. So this is very important and related to the informational structure. So the Oizumi talked about the relationship of the neural activities, information, and the consciousness of something. This functor itself is some kind of mapping. And the natural transformation is a relationship between these mappings. The natural transformation itself is a family of arrows in D, category D. So like the TX, TY, that is a mapping from each object in C, category C, to the arrow in D. And making this diagram commutes. So this is the notion of natural transformation. Then what is natural equivalence? This is a reversible natural natural transformation. So this TX is if reversible, this natural transformation is called a natural equivalence. If the two functors have linked by natural equivalence, mathematically the functor can be essentially the same. So what is the functor categories? So we just summarize the notion of category and the category of categories and functor categories. Let's see these big ellipses have the categories C, D, E or something. Of course, we can consider the category of categories, that the objects are categories and the arrows and functors. But the interesting thing is there is more meta structure of the category of categories, that is functor categories, that between two categories, we have another very rich category that functors are objects and the natural transformation is the arrows. The Yoneda lemma says that any categories can be embedded in this kind of functor categories that like the object functors and arrows and natural transformations. So let's say the category of mobility, of course, can be considered as functor category by Yoneda embedding. Any localism is a very technical subject, so please ignore it. Any categories can be embedded in the functor category. This is Yoneda. And the objects are isomorphic if the corresponding functors are naturally equivalent. This is Yoneda lemma. Most interesting category, and also the most interesting categories are originally arisen as a functor categories. In physics, for example, representation category, that the group that is some kind of group like a Poincare group in relativities. The arrows intertwines between. This is a particle physics is all about this category of rep G. And the category of mobility, it's self-state as a functor and state transition as natural transformation. Because Yoneda lemma, Yoneda embedding, we can consider the state as functor and the state transition as natural transformation. But as the same as the representation G, rep G in physics, this, my speculation is originally the category of mobility for the consciousness studies is originally maybe the functor can be considered as functor categories. That from neural activities, information, and the consciousness, that functor itself can be considered as diagram, because you can see this functor can be considered as embedded diagram of C into D. So some embedded diagram can be considered as functor. So some patterns can be considered as functor, and the pattern changes. The changing diagram is considered as natural transformation. My very speculative feeling is that maybe the contents of consciousness or qualia is related to this kind of natural transformation changing diagram. Okay, so far we focus on the qualitative structure of category, because basic category theory is all about a very qualitative thing. But of course, as Oizumi stressed, that we need a quantification. The quantitative structure is we need. So how we unify the category theoretical, very good qualitative structure with a quantitative structure. So the basic strategy is to consider the enriched category. What is enriched category? So for example, for the case of the category mobility, dynamics as development of weight of arrows, that the category to consider the weight, we need more structure on set of arrows. If the set of arrows are structured in some sense and the structure behave in harmony with composition, then the category becomes enriched category. So we introduce what is enriched category in the next slide. Oh. Sorry. Okay. Simply put, an enriched category is a generalization category by replacing the notion of set of arrows with more structured, like a quantitative structure for the set of arrows. So enriched category is a set of objects equipped with a generalized set of arrows as an object in a monoidal category. So sorry for this short lecture, so I cannot explain fully, but the point is, enriched category is a category with quantitative structure, like arrows has some weights or something. So by this weighted arrows, we can use enriched version of the Yoneda approach. So this is an ongoing study by Tsuchiya, Phillips, and me. And a related new approach is using category algebra. The category algebra is some kind of polynomial, generalized polynomial algebra generated by arrows as indeterminates. As polynomials, we can make the coefficients of arrows, and we can some kind of substitute something. So this is essentially a convolution product for this arrows of arrows. So multiplication reflects the structure of the composition of arrows as convolution, and the category algebra becomes non-commutative algebra. So this is related to the quantum probability approach for the origin from physics, but Makiko and collaborators also studies about this quantum probabilistic structure of consciousness. So I believe this category algebra approach is related to that kind of study. And the state can be defined on the category algebra as a linear function on the category algebra. The combining the network theory to the category theory, information theory, and I believe that this category algebra approach will be contribute the how quantitative structure, how we focus on the quantitative structure, not only the qualitative structure of consciousness. So thank you very much for the listening.
Thank you.
Yeah,
so that was great. I think we have time for a discussion now. So we have about 15 minutes. And I don't know if there's questions, so I don't see any questions at the moment in the Q&A or chat. Or raise
so maybe if not, then or I or Makiko can start. Makiko, do you have any question? Or if not, then maybe I'll start. So
yeah, that sounds good.
Yeah, okay. So that was great. Both of the talks really clarified what we want to do in a Qualia structure. Hopefully, also to understand to be understood by Marieke. But I thought that it would be also good to point out what Hayato wanted to also deliver as a sort of the natural transformation. I mean, just to confirm what you said, right? So usually in neuroscience, including both Makiko and also Marieke's study, the state of the neural populations measured by fMRI or neural firing and so on, that's regarded as a sort of static number at each location, right? And then that either average over time or average over the space, that's considered as almost like object category theory, right? And then the relationship between these are, in a sense, kind of a correlation or something like that. But what you are saying is like not only this kind of momentary activity state as an object of the study, but using category theory like idea, how these patterns interact from time 1 to time 2.
Yes, exactly.
This itself can be the study of the study object, and you think that that's correlated with the Qualia.
Yes. Exactly.
And that I think will relate to what Masafumi was saying as a sort of IIT is a theory or proposal to compute a causal structure.
Yes.
Right. So that also deviates from the standard neuroscience where, state as a sort of static thing, but it's actually how the system changes from one time to the other.
Yes, exactly.
Okay.
Yeah, that's definitely helpful to understand it better, and it is indeed introducing in some ways dynamic, right? In saying the brain also is a dynamical system, and the current state can be maybe predicted by the previous state, and some transformation apply to that previous state. So is it indeed, so do I understand correctly? It's related to that, and that you could potentially think of these transformations between states, in the way you describe. And then you could do this, and then there's the higher level, the meta level, where you can do this both in terms of measuring the brain activity and measuring somehow your perception or qualia, conscious qualia, and then relate them to each other, right?
And the related point is that in my presentation, maybe the many audience can see that there isn't anything like a network relationship between network theory or category theory. Many can ask the category theory is a part of network theory or something, or the answer is the conventional network theory is some kind of the horizontal network that is in some level we can say some network, but category theory is not only horizontal but also vertical structure beyond hierarchy, because very higher kind of networks can be considered as an object as an arrow. So we can translate this hierarchical structure of networks. So in that sense, category theory can include conventional network theory and has many regions which conventional network theory does not focus on so far, have not focused on so far.
Yeah. And also, maybe if there's any further questions, if not, then maybe I'll,
Yeah, go ahead.
Marieke.
I have another question later, which is more related to Masafumi's talk.
I just also wanted to point out that, towards the end, Hayato was talking about the enriched category, and that's a bit complicated. And I also initially didn't really understand it, but it's like easy way to think about it is like not to consider arrow as whether there is present or not, but it has a degree, right? Like, like we are measuring similarity is a kind of, degree of the arrow. And to deal with this kind of arrows, then we need to go beyond standard category theory, but also go to enriched category. And then with that, we can do kind of combine all these similarity rating, derived structure, and sort of a neuronally kind of stochastic network probably, right? So far,
can I ask, so you are saying that the enriched category is something like a weighted graph, or not a binary graph?
Yeah, this kind of weighted graph, from the weighted graph, you can construct some enriched category. But the enriched category itself is a more general thing. So first we try to understand a very simple enriched category called the generalized metric space. The which is one of the simplest example of enriched category, which is a generalized metric space means that the same thing has a distance 0 and some kind of the triangle inequalities. But we don't need symmetry of the distance. Or we allow, for example, infinity, the very, very dissimilar or something. So this generalized metric space is in some region is very fit to the experimental test, I think.
I see.
And that also relates to what I mentioned in the beginning or last discussion, right? Like similarity may not be asymmetric perceptually. And so, enriched category allows this kind of thing as well. But also, I think, Hayato, I also was interested or, impressed by this possibility of the category algebra that allows you to kind of think about a network that has a loop and also multiple pathway. Which is probably, Marieke, you're Makiko, you're the neuroimaging expert. So you probably, have you encountered any kind of graph theoretical analysis that allows that, let's say node A and node B is connected in two ways, or it also has a unidirectional or asymmetric kind of connectivity strengths, or while allowing also self-connection kind of thing?
Yeah, that's an interesting point. So I haven't really done a lot of connectivity analyses myself, but the standard analyses do not allow that. They just, you know, it's one, often people just have time courses of nodes, right? And then they correlate them, and that's the connection. And it's just one correlation value. I'm sure there is more advanced methods, but I'm not sure if they allow for that. And I think in terms of thinking about modeling, there is maybe a little more I can say if you think about these neural network models of, for example, the visual system. They're only recently, in the last few years, I would say, there is a consideration of including recurrent connections between, for example, nearby units, or also having feedback connections. So there you would indeed, for especially the feedback, you can have different weights for going from, for example, V1 to V2, then from V2 to V1. So that's starting to be incorporated in these networks. And now you get indeed networks that are universal approximators of dynamical systems. So that's kind of interesting. But yeah, in terms of the actual analyses of fMRI data, it's not standard. That I definitely know, to look at these multiple connections, and that they could be different, right, in terms of their weights. Yeah.
Okay. Did you guys want to answer, Hayato or Masafumi? Otherwise, maybe Marieke, you had also one question, right?
Yeah, there was one thing just to better understand in terms of what to me was quite interesting was the part where you have developed these methods to identify, let's say, the region, right, region A. I was wondering if you could explain a little bit more about how that works. So how do you determine which subsets, let's say, of nodes, right, maybe form this complex, locally most integrated system?
So we have some way to quantify so-called integrated information in a particular subsystem. So in this case, the whole system is something like this, and we quantify the integrated information in each subsystem, like this one and this one and this one. Many, possible subsystems exist in the whole system. So we basically all calculate all integrated information in all possible subsystems, and then we detect the maximum among all possible subsystems. Then we identify the maximum as a complex and most integrated subsystem. But this is computationally basically intractable in large system. But we have some algorithm to deal with this combinatorial problem, and then we can find this complex exactly in several hundreds units network. In this case, actually, that we have 128 electrodes in ECOG data, and then by using all of the channels, we detect the complex or most integrated subsystem, something like this. So the posterior part of the brain corresponds to the most integrated subsystem in this case. This kind of thing can be done.
Yeah, that's interesting. It's so then if you think about applying this to human data, so imagine now you have fMRI data. I guess the initial approach would then maybe be to look at, let's say, parcels or regions, right? That you have a reasonable number, maybe a few hundred max, and then look at that. So would that indeed be something that you think that should be feasible?
Yeah, it's feasible. Actually, we are doing also a functional MRI analysis as well with the same method. And we,
that's nice.
We can also find this kind of posterior part as a complex in even functional MRI data.
Oh, that's nice. That is consistent then. And then between the monkey ECOG data and the human fMRI data. So then, how would you interpret this as is that where consciousness resides?
That's a difficult question. So just by using the neural recording data, we cannot say anything about consciousness.
No.
My proposal here is that we need to connect this with psychophysical experiment. I mean, Qualia structure. So the hypothesis here is that if this hypothesis is correct, then this part should explain the Qualia structure. I mean, the activity in this part should most explain the Qualia structure. I mean, quality of consciousness. If we can test that thing, we can say something about consciousness. But not only from just computing the complex from the neural recording, we cannot basically say anything about consciousness. What we can say is just this is the most integrated subsystem. This recording. That's it. So.
Yeah, but that's definitely a very good starting point. I think it's great. Yeah. Okay, we have to move on. Actually, we're already at the end of our discussion time.
Yeah.
So,
thank you for the question. Yeah.
Yes, thank you to all the panelists for the very interesting discussion. So I think, and now we have only one speaker left, right? Which is now. The title of the upcoming talk is Massive Report Paradigms to Understand Qualia Structure. And this is just to read the number again, right? Make sure that everyone knows which, it's 1S09M. So that's our symposium, and then it's the 5th talk. So the floor is yours.
Thank you, Marieke, for co-organizing this symposium. And I think it has been quite a good, coherent symposium, even though it has been very interdisciplinary from imaging to mathematics. And then I'll try to wrap up this with my massive report paradigm talk. All right. So the dominant paradigm of consciousness research over the last 30 years has been to trying to identify the brain areas which correlates most with the subjectivity or conscious perception of something. Like, when you are seeing dog or thinking about dog or whatever, then a dog neuron in the brain are supposed to be firing. But that's also, a kind of a potential bottleneck of this type of the study. Can we expect actually a fundamental shift in understanding neural correlates consciousness by just extending this, NCC approach? That's the sort of the first question we started to think about. And then our answer is maybe not. And one of the reasons is that, as alluded by Masafumi, categorization of experience into, let's say, seeing the dog or not, or seeing red or not, or can you just discriminate between red and purple, is definitely not enough to capture what we are actually experiencing. And one really need to have is this, this establishment of the method to deal with quality of experience, or what we are actually seeing right now, or experiencing, hearing, or feeling, and so on. And the one way to quantify Qualia is to characterize the massive relationship among Qualia, which has been already discussed in Hayato and also Masafumi's talk. So I'll be very brief. And this is indeed, inspired by Yoneda lemma. And the Yoneda lemma says that, even if you cannot say exactly that, red is what the red is, but through the relationship with other colors, you can identify what the red is. That's a sort of the inspiration from Yoneda lemma. And exactly whether this actually applies or not, mathematically, is something that I and Hayato has been working on for a while. But that's already good enough as a sort of inspiration. We don't need to really care about exactly what the red is, like philosopher does. But if we just understand the relationship, like my relationship with other people will define me, it is enough already as a starting point. That's the idea. And then we're using these ideas. I'm going to talk on 2 topics today. First is that just starting with the color, like Makiko initially talked about. Do we see colors at the fovea and the periphery in a similar way? It's some kind of, for those who are not in psychology, department and so on, you may say that, oh, this is so, true that, I see color at the fovea and the periphery, no problem. Why are you asking? But in fact, if you open up the, psychology textbook, this is one of the first things that you learn that it is not the case. And that's something that we want to address first. And then the second thing is that another sort of textbook kind of statement that when you see a scene or briefly, see some kind of, scene, movie scene, whatever, what you are experiencing is only a gist of a scene and not really the details. And that's also another kind of textbook kind of concept, which has been not, asked as a sort of a solid kind of, gold standard in psychology. But is it actually true? That's the question. So in experiment 2, I'm going to ask about, what is the structure of a qualia in the moment of experience? So the key to address this question, and also in the reason why psychologists has not been able to directly answer this, in my opinion, is that they have been, in a sense, confined in a mode of the thinking that, we need to carefully ask the reports and so on, and then, you need a controlled stimulus and so on. But if you come from more, a radical kind of approach, more stimuli, more relationships, and they're giving a freedom to the participants, that's the key to do this kind of thing. All right. So the first part, do we see colors at the fovea and the periphery in a similar way? And as I said, if you take the psychology 101, what you learn as one of the first lecture is that the density of cones and the rods are very differently distributed in the eye. And the cones are supposed to be critical for seeing the color. And the rods are blind to the color, or wavelength. And as you can see here, shockingly, that cone density actually decreases really dramatically, except for the point of the fixation that you're looking at exactly. And it almost goes like, looks like 0 right after 5 degrees or 10 degrees after fixation. And what it means in terms of the actual display is something like this. So if you hold your finger in front of your display and then adjust your size like this, for me this works really well, then that fingernail of the thumb roughly corresponds to 1 degree of visual angle. And that means that this red square area is already corresponding to this, cone, relatively rich kind of area. That means that all of your sensation of color here is coming from this, stupid, areas of the, color, low color cone density kind of, areas. And if you are like me, you are claiming that, we do, see color. But, with this, sparse color cone, how it's possible? That's a question, right? And many philosophers and also psychologists have argued that, what we are actually seeing in the periphery color is just an illusion coming from expectation, memory, and so on, magic, basically. Or they might even say that Qualia, study is just impossible, as this, demonstrates that, it's just unreliable. Our introspection is just unreliable. Stop doing the consciousness research. That's a kind of a claim. And what Ariel Johnston in my postdoc has started is asking this question. Okay, let's massively, just characterize the relationship. To our surprise, most of the color study has been always performed within the fovea, mainly probably because they believe that cones are only present around fovea. So it's like comparing red and green here. And then we use a maximum display size to ask the relationship between this and this. And then in collaboration with Makiko, we are testing this far periphery as well. All right. So this is the example task structure. So we just showed 2 patches at a time and then asked how similar they are, from 0 to 7. So this is very, easy thing. And according to some kind of psychophysics, then the only thing that you can reliably see in terms of the color is when things are presented at the fovea. And here we don't have any expectation, any limitation in reports, and then you can just, reliably, just tell what they say, right? What do you see? And then what we found here is that essentially the similarity relationship structure at the fovea and also periphery is basically the same. That's the first thing. And the secondary, central color and the peripheral color are matched, almost like, predicted from central or peripheral prediction. So this is not exactly the kind of existence proof, but I think this is sort of similar to functorial kind of relationship that Hayato said, right? There's a relationship within the fovea, relationship within the periphery, and these relationships are preserved by mapping from fovea to periphery to fovea, right? So that's sort of the equivalence of the color experience in the fovea and periphery. And now we are our paper is under review but Makiko and I, we are testing whether this also happens at the even very far periphery using this new device Makiko has. All right, so that's the first component. And then the second component is that can we experience more than a gist of a briefly presented photo? And you might have seen this, another psychology 101 kind of stimulus called Neibon stimuli. And in Japanese, sometimes called, kiyomite moriomizu. You see either local things and then don't see the global, or in this case, most people see only the global, but not the local. Here is another kind of demonstration, but you see like a natural scene on the left side, but if you look at the local details, then it doesn't actually make sense. It's a half kitchen. And some image manipulation says that even if we degrade periphery, some people just don't notice whether the periphery is degraded or not. And also it's related to the neuroscientific finding that there are some neurons that respond to the global property of the picture, but not the local. And the local things just comes much weaker and also later. So all of this kind of suggests that upon briefly seeing a natural scene, what we see is only the global and the coarse gist. And if you are like me, and then if you see something that is very detailed and concrete, that's an illusion again. Okay, that's another philosopher's topics. But let's before concluding, let's check our own phenomenology. And if you can type in your chat box, please type in what you see in the next image in chat box, up to 5 words, please. Okay. Are you ready to do this? Okay. 3, 2, 1, 0. Okay. So please type in what you saw in the image. Now the time is over. Okay, it may be a bit difficult, but anyway, if you're like me, then I think you probably saw some kind of outside scene, and it was probably like Eiffel Tower, and it looks like sunny and park, and then there is some the tower on the left side and the trees on the right side and so on. But this kind of thing, the concrete feeling of this Eiffel Tower is too specific or locally detailed compared to the common sense. And where does it come from? And it is, my experience was at least structured and spatial arrangement was there, and it's not like really blurry kind of impression of the outside or blurry kind of parkness, right? It was more to do with the concrete object over there. And if this is possible, maybe it's that the previous conception that we see only local, global and coarse thing is a limitation that experimenter imposed on the participants by not allowing them to report what they saw, and that's the hypothesis that we posted like 4 years ago. And then to address this question, in collaboration with these students and also Yusuke and so on, studying in children, we are trying to remove this experimental's bias. And in bias, what I mean is that, in the case of even similarity, we already tell them that we are going to present this stimuli. And in the case of color, this color. But if you don't have any expectation, and also if you allow people just to type in what they see, then there's absolutely no, limitation. And then let's see how it goes. But then the problem is that you can't score anything, And our radical conception is that, let's forget about what is the right answer. Consciousness doesn't have a right answer from the experimental point of view. If you say you saw an Eiffel Tower, that's correct. And then let's ask what others say. So that's a majority vote rule. And let's see what I mean. And then this is only possible when you test number of massive stimuli and massive trials and massive participants online. And that's what we did, basically. So in each trial, we asked them to type like 5 words, like I asked you. And then we tested like over 600 people and then 400 images. And then this is the image that I showed to you. And then in the case when we presented relatively long, that was the 10 people's response of the 5 words, and 133 milliseconds and 67 milliseconds. And common sense says that 67 milliseconds, you can only see like blurry color and relations of the blurry object. But that's not the case. In fact, to test the specificity of this particular report of the Eiffel Tower, what you can do is to see whether other people also report that. And in fact, in this case, 13 out of 20 other people reported Eiffel Tower. And in fact, for none of other images was reporting, nobody reported, Eiffel Tower. This means that, report of the Eiffel Tower is highly selective and also specific, but it's commonly shared across many people. And that we can quantify it as an intersubjective agreement score of 1.0. And using this method, what we found is that the different people seeing the same thing can report different words. Okay, that's fine. We don't report everything in a similar way. However, when you report it, it is really specific to that particular image. And you don't use that word for other images. So the median intersubjective agreement is really high across all the frequency of the words reported. So in summary, I just wanted to say 2 things today. Qualia structure between fovea and periphery is that it's more or less equivalent. And the Qualia structure on the briefly presented natural scene is that it's actually locally quite detailed. So the next step, naturally, is that how can we model these results? Like moment of the experience is highly rich, and then it's somehow kind of supported by the brain and the information. Can we do that? Model that from a category theoretical point of view? And also, what are the underlying neural mechanisms and the information structure? So that's the next step. Thank you for your attention.
Yeah, thank you for that exciting talk.
At least I was excited.
Yeah, it's definitely very interesting. And I think these are, as you started, 2 sort of common conceptions and in psychology, and you're testing them, and actually they don't seem to be holding up completely, right? Which is nice. So yeah, we have some time. So we have 6 minutes, I think, for further discussion. So I don't know if there is if anyone has questions. I am not seeing anyone here online. I do have one question that I'll be happy to start with. Because one of the things that's interesting about when you say GIST, I immediately have to think of this model, right, of Oliva and also Taroba, where they have these, they call it the GIST model, right, where you just basically can put images through their model, and what's being extracted is different orientations, information at a range of spatial, I think, frequencies. And they say, like, when you extract that, that's considered the gist of the scene, because that's something that could be extracted quite quickly. And I think they have, of course, some data also from humans to sort of support that idea. So I was wondering, what if you would, that's just one option, right? But the images that you have, if you would pass them through their model, it's relatively simple. It's a bit like a V1 model, but at multiple spatial scales. And that's and if you then after that would, for example, see what people could still recognize, or whether you can measure somehow the presence of these different types of information in the image, That would be useful. Yeah, some way of having like a representation of the gist of the image and then see what's present and whether people mention more local things than that. This is sort of more general idea, right? And I'm not sure if the gist model necessarily would work, but that could be potentially interesting to see if they as a sort of baseline or something. I don't know if you've thought about these.
Yeah, so we haven't done the exact kind of model study, but this is probably quite an interesting data to share. So this is a subset of the data that we have, and as you know, we have this kind of thing for 400 each of the 400 images, and from this alone, I feel like it's highly unlikely that their gist model will tell us anything like this. For example, this, for example, I was quite surprised actually when we show this, letter array, sometimes using psychology experiment. I thought that many people would just say like a letter, text, alphabet, or something like that.
Right.
But in fact, they do report like U, top left. It's definitely specific, right?
Yeah, that's
and
true.
I'm not sure, whether we can get any kind of orientation model to just get to the U among all of these natural things, completely unexpectedly presented. If you have an expectation, yes, you can.
Another question now that brings up, basically what you're explaining now brings up another question. So in terms of people's eye movements, I know you can't make many eye movements, right, in a very short amount of time. But are people looking at different parts of the image when it comes up? Do different people look at different?
Yeah, so that's actually really excellent question, because I was also very surprised that people report from top left, right?
Yeah.
And I thought that that's probably what you're saying. So even though the fixation cross was here, 10 out of 30 people reported a U. And then we also have a black background and the white letters condition as well. And then there again, top left letter was reported most. And my guess is that this is interesting kind of thing to follow up later. But after seeing this kind of array of letters for 67 milliseconds, your mind eye, in a sense, probably scan from top left.
Yeah, that makes sense. Probably. Yeah.
Can I ask, is it only for letters, or is it all goes for the other scenery pictures?
So if you see like here, the apron here is artophobia, right? And it's not like top left thing like shelf is reported here. It's a daughter, here and so on. So most of the time, people do report most salient thing actually.
Yeah.
And it's not like predictable from just a pure location. And these are other
Yeah, because I was wondering, because in our writing system or reading system, it goes from left to right.
Right.
So if it might be like related to cultural things, like.
Yeah, I think so too. And that's probably also part of our structure of consciousness, in my opinion. If you are, raised in certain kind of, culture, your reading system will, shape your experience like this way. I don't have a good other example of top left reading, but testing in Israeli population could be one possible way to, address this kind of question.
Yeah, that's very interesting. It's we're already at the end. I think we need to do the closing statement.
Oh yeah.
I don't know if you want to do that. 2 minutes left.
Okay. I think we are exceptionally good at timing.
Yeah.
All right. Thanks, Marieke, for the great chair. And I didn't prepare much of the closing remark. I'm just sharing this slide with all the participants for those who are still here. And if you are interested, you can just go to some of these URL to follow up the discussion and the question. And we are also planning to have a follow-up discussion, at least between me and Masafumi, and maybe a bit with Marieke. Unfortunately, Makiko and so Hayato needs to go somewhere. But yeah, so that's the thing. And just to remind you, we will have put this talk parts into YouTube. And also the future plan of the symposia include development of consciousness from infants and also babies. And we have some YouTube channels and also Twitter account for me and Masafumi and Yusuke. All right, that's it. Thank you very much. And especially for those people who are up until here. Do you have any further question or comments among you all panelists? Thanks a lot.
Yeah, thank you. Thank you to everyone. It was very interesting.
Thank you. And Marieke.
Thank you so much.
Thank you.
Thank you for organizing.
All right, great. So if you are interested in going into YouTube, that's this here. So, all right, then, yeah, you can leave, or I can already start this thing. Oh. All right, maybe we can stop recording for Zoom. Well, maybe it doesn't need to. I don't know.
So thank you very much.
Very much.
Thank you.
Bye.
Bye.
Yeah. All right. Bye bye.
So for YouTube, where should we do?
I mean, we can just continue talking here,
yeah,
and I'll just start broadcasting. Yeah, thanks, Marieke, for the great host and also questions and so on.
Yeah, same. Yeah, I thought that was certainly nice to see these different, indeed, work at these different, in the different fields, right, that we're trying to sort of integrate neuroscience, mathematics. And it's nice to think about that in a, also the mathematical aspect, which is indeed, for me, not something I usually think of as much, right? I'm doing first experiments. I try to characterize the representation and relate them, but it's nice to think about it at a little bit more abstract level and think, how can we actually test these different theories of consciousness? Because they do seem a little ungraspable, right? If you, so